Priyana the science girl. Priyana the science girl. Priyana the science girl. Science rules. Priyana the science girl.
Inertia is a property of matter.
Science girl. Priyana the science girl. Whoa, I didn't see you there. How are you?
Get off my screen!
You said you want to learn the cell cycle? Mitosis? Alrighty, I'll teach you all about it. Let's get on. So, let me teach you all about the cell cycle. The cell go through a cycle, and that is the cell cycle.
You think?
So, here we go. The cell cycle. What do they do? Cells do three main things. Okay, got it? Good?
I get it. Now get off my screen.
They, number one, they grow. Number two, they perform the normal, I believe that's the word, function. And number three, they multiply. Let me show you how. We have grass right here. Alright? Now we have all this grass. And that's how they multiply. Now you guys are still probably confused as to how a cell goes through the cell cycle and what happens in the cell cycle more specifically. So now we're going to have Tea Talk with Karen. Hi Karen, thank you for meeting with me today.
My pleasure.
Okay. So, tell me, what is the process of going through the cell cycle?
Okay, I found this on the web for what are the 5 stages of the cell cycle.
Sorry, I'll try harder.
Okay then, Karen. Sorry.
Oh wow, interesting. It's interesting. Thank you. I will elaborate further on that. The tea has been spilled.
Okay, bye.
So, now that Karen has told us the process, right Karen? Now that she has told us the process of how the cell the cell cycle, I will also tell you the process of the cell cycle. So, let's get on with it. Bam! You can see, but bam! Last but not least, bam!
ASMR time. Settle in, kids. The cell cycle. During the cell cycle, cells grow, cells perform their normal function, cells multiply, and there are 5 stages in the cell cycle. 4 of those stages are in the mitosis. What is a mitosis? The process of how diploid cells divide. What is a diploid cell? A cell with paired set of chromosomes. For example, human cells contain 46 chromosome cells. Die. Evidence: dust. Dust is made from dead skin cells. Mitosis is dead. JK. Important. Mitosis become new diploid cell to replace cell that die. Interphase. First stage of the cell cycle. 75% of the cell cycle. The interphase stage has 3 stages within the stage of the interphase stage. First stage within interphase stage is death. JK. I need to go home. The first stage of the interphase stage is G1. G stands for growth. What happened in G1? Cell prepare for mitosis, meaning it grow and begin to create more organelles like vacuole, lysosome, smooth emergency rooms, ribosomes, and Golgi bodies. Let us demo. Here we have circle within circle, regular schmegular cell. We have ribosome, smooth ERs, Golgi body, lysome, vacuole, nucleola, and nucleus. You see, circle and circle. And then it get bigger, the cell swell. So we get a tiny cell to a big cell. Random question quiz. What would a stomach cell do during interphase? Stomach cell would release enzyme to break down food. The second cell of interphase is S, synthesis. S mean to make. During the S stage, the cell make extra amount of DNA. DNA is currently in form called chromatin. Chromatin, long loose strand of DNA, like spaghetti. Chromatin, spigo. During S, the double helix split in two new pieces of DNA are created. See? DNA uncoil and two new pieces. Third stage of interphase is growth 2. For the second time, cell will grow in size and become larger. So we go from G1 to S, which means more DNA, and G2, which is even bigger than G1. So the cells swell even more swell. Random question quiz. True or false? Interphase is a quick stage in a cell's life. False. Interphase is not a quick stage in a cell's life. The next stage of the cell cycle is prophase. This is where mitosis begins. At the start of prophase, DNA is a chromatin. It's a spigo, remember? During prophase, DNA began to coil into easily movable structure called chromosomes. Here we have a chromosome. What is the purpose of DNA? Coiling into chromosomes from chromatins. This is why. Because it's easier to transport to other cells. Remember, it is a suitcase, okay? Nucleoli. Purpose of nucleoli is to store and protect DNA. However, it is in the way. During prophase, the nucleus will devolve. How? Enzyme present in the cytoplasm will actively break apart the nucleus. Here we have a nucleus breaking apart. Spinal fibers from during prophase. Spinal fibers from during prophase. Spinal fibers are created by organelles called centrioles. See, we have a centriole and spinal fiber. Third stage of the cell cycle is metaphase. Spinal fibers create and prophase. Remember, what are they? A long thin thread of protein that grow from organelles called centrioles. So here we have the centromere and chromatid. So during metaphase, spindle fibers will attach themselves to the centromere location of each chromosome. Centromere is a location where two halves of the chromosome overlap. Chromatids, the two halves of a chromosome. Once spindle fibers attach themselves to the chromosomes, spindle fibers will pull and tug on the chromosomes until all are aligned in the equator, which is in the middle of the cell. See? Chromosomes aligned in the middle of the cell. The fourth stage of the cell cycle is anaphase. Spindle fibers are going to continue their tug of war. You see? During anaphase, spindle fibers are going to pull at the chromosomes in opposite directions. See? 4 chromatids to the right and 4 to the left. For human, 46 chromatids will pull to the right and 46 to the left. Anaphase, chromatids separating. The fifth and final stage of the cell cycle is telophase. The nucleus returns, so you have the nuclei forming. During telophase, the cytoplasm divides. See, you got a butt. Remember a butt. And this is called cytokinesis. DNA is a chromatid in the beginning of telophase. Then it uncoils into a chromatin. See, goes back to spigo. And then we have 2 cells. Wow, look at that, 2 cells. We start from 1, now we end with 2. How cool. Remember, IPMAT, I is independent. Don't go with that. Okay? From math, that means mitosis. Okay? Remember, I independent. Why should we care about mitosis? Cancer. The number 2 cause of human death is cancer. Cancer cells are errors that prevent the shut-off of mitosis. Cancer cells are basically stuck in mitosis. The understanding of mitosis helps to save millions. Cancer cells are constantly dividing by mitosis. As they multiply, eventually a lump of cancer cells begin to form. This is called a tumor. Tumors can be harmful because often cancer cells can metastasize. This means a few cancer cells can break away from the tumor and travel through the bloodstream and into another part of your body, making treatment difficult. See, right here we have metastasizing. Cancer cells break through the basement membrane, and then the cancer cell detaches from the tumor and can spread to other parts of the body. Then we have 100 trillion cells make up the body. So, there's no easy way to find and kill all of the cancer cells. Cancer cells can be targeted by X-rays. However, the radiation also kills normal cells. This is radiation. Goodbye.